Today we will talk about a new topic, the cell cycle. The key roles of cell division, the ability of organisms to produce more of their own kind, best distinguish living things from non-living matter. The continuity of life is based on the reproduction of cells or cell division. In unicellular organisms, division of one cell reproduces the entire organism. And multicellular organisms depend on cell division for development from a fertilized cell, growth, and repair. Cell division is an integral part of the cell cycle, the life of a cell from formation to its own division. On this slide you can see three figures, figure A, B, and C. On the top figure, figure A, you can see a diagram of reproduction of amoeba. As you know, amoeba is a protozoan. In figure B, you can see growth and development of a sand dollar embryo. And figure C shows you tissue renewal in dividing bone marrow cells. Most cell division results in genetically identical daughter cells. So, result will be in daughter cells with identical genetic information which is located in DNA. The exception is meiosis, which is a special type of division that can produce sperm and egg cells. More deeply, we will talk about meiosis in the lecture of general embryology. Cellular organization of the genetic material. All the DNA in a cell constitutes the cell's genome. A genome can consist of a single DNA molecule, common in prokaryotic cells, or a number of DNA molecules, common for eukaryotic cells. DNA molecules in a cell are packaged into chromosomes. In this figure you can see chromosomes. Here are they. That's a genetic material, and that is a cell. As you consider, that's very large cell. Eukaryotic chromosomes consist of a chromatin, a complex of DNA and protein that condenses during cell division. Every eukaryotic species has a characteristic number of chromosomes in each cell nucleus. Somatic cells, or non-reproductive cells, have two sets of chromosomes, or full number. Also, they are known as haploid. And gametes, which are reproductive cells, these are sperm and egg cell, have only a half as many chromosomes as somatic cells, or they also known as haploid. Somatic cells are diploid and gametes are haploid. Distribution of chromosomes during eukaryotic cell division. In preparation for cell division, DNA is replicated and the chromosomes condense. Each duplicated chromosome has two sister chromatids, joint copies of the original chromosome, which separate during cell division. The centromere is the narrow waist of the duplicated chromosome, where the two chromatids are most closely attached. Here we can see two sister chromatids and centromere in the middle of them. During cell division, the two sister chromatids of each duplicated chromosome separate and move into two nuclei. Once separate, the chromatids are called chromosomes. So before separation they are called chromatids, after separation they are chromosome. So here on this figure you can see chromosomes and chromosomal DNA molecule. Here that's one arm of chromosome, here is centromere. So when chromosome duplication occurs, including DNA replication and condensation, here we have two sister chromatids with double number of DNA, and how it really looks under electron microscope, under transmission electron microscope. And when they are separated, separation of sister chromatids into two chromosomes here, they become chromosome. Before they are called chromatid, joined by centromere. Eukaryotic cell division consists of mitosis, which is the division of the genetic material in the nucleus, and cytokinesis, the division of the cytoplasm. Gametes are produced by abbreviation of cell division called meiosis. So gametes are sex cells. Meiosis yields non-identical daughter cells that have only one set of chromosomes, half as many as the parent cell. So before I told that they are also known as haploid number. The mitotic phase alternates with interphase in the cell cycle. In 1882, the German anatomist Walther Flemming developed dyes to observe chromosomes during mitosis and cytokinesis. Phases of the cell cycle. The cell cycle consists of mitotic or M phase and interphase, which is the cell growth and copying of chromosomes in preparation for cell division. Interphase takes about 90% of the cell cycle, can be divided into subphases. G1 phase, which is also known as first gap, then S phase, S is for synthesis, and G2 phase, which is second gap. The cell grows during all three phases, but chromosomes are duplicated only during the S phase. Here on this diagram you can see all the phases. So we told that 90% of all the cell division is for interphase. Interphase includes G1 or gap 1 synthesis phase, where DNA is synthesized, and G2 phase or gap 2, and mitotic phase, which is marked by letter M, where first mitosis occurs and then cytokinesis. As a result, we have two cells. So from one cell, second daughter cell is appeared. Mitosis is conventionally divided into 5 phases: prophase, prometaphase, metaphase, anaphase, and telophase. Cytokinesis overlaps the latter stage of mitosis. Here on this slide you can see all these phases, including G2 of interphase. In the top part you can see a real diagrams of these phases. In bottom slide or bottom figures you can see schematical illustration. So let's start from G2 interphase. Centrosomes with central pairs and chromatin is duplicated in this phase. So in prophase, early mitotic spindles form and asters of centrosomes are formed. Also, genetic material is attached by centromeres. Chromosomes consisting of two sister chromatids. Here we have. In a stage of Prometaphase, fragments of nuclear envelope appear, and non-kinetochore microtubules also appear. And as you notice, centromeres, they move into opposite poles of the cell. And here also kinetochores form, and kinetochore microtubules. In metaphase, spindle is formed, and metaphase plate is also formed, and centrosomes also appear at one spindle pulse. In anaphase stage, daughter chromosomes, they are separated, so, and kinetochores and microtubules start to move towards centrosome chromosomes. Telophase and cytokinesis stage begins by forming a cleavage furrow. So, in animal cells, cleavage furrow forms. In plant cells, cell plate is formed. That's a division line. And also, both cells, original cell and daughter cell, they start to form their nucleolus and nuclear envelope. The mitotic spindle, a closer look. The mitotic spindle is a structure made of microtubules that controls chromosome movement during mitosis. In animal cells, assembly of spindle microtubules begins in the centrosome, the microtubule organizing center. The centrosome replicates during interphase, forming two centrosomes that migrate to opposite ends of the cell during prophase and prometaphase. An aster, a radial array of short microtubules, extends from each centrosome. The spindle includes the centrosomes, the spindle microtubules, and the asters. During prometaphase, some spindle microtubules attach to the kinetochores of chromosomes and begin to move the chromosomes. Kinetochores are protein complexes associated with centromeres. At metaphase, the chromosomes are all lined up at the metaphase plate, an imaginary structure at the midway point between the spindle's two poles. Here you can see the formation of aster. Aster is a radial structure with the centrosomes in its middle. Also here you can notice kinetochore microtubules. Here it's shown in schematical illustration, and here you can see it in a real transmission electron micrograph. And kinetochores. Here is overlapping non-kinetochore microtubules, and that is metaphase plate, which is imaginary. And here also that's a transmission electron micrograph. Here you can see a lot of microtubules with chromosomes and with centrosomes. In anaphase, sister chromatids separate and move along the kinetochore microtubule toward opposite ends of the cell. The microtubules shorten by depolymerizing at their kinetochore ends. So, that is very interesting diagram showing an experiment. Here on this experiment you can see kinetochores, and that is spindle poles, one in that side and one in opposite side. A middle portion of microtubules, of kinetochore microtubules, are marked, and as a result you can see that microtubules, when they are depolymerized, they start to shorten and move toward a spindle pole from that side and from opposite side. As a conclusion, centrosome moves towards a spindle pole, and here we have motor protein, kinetochore, and when there will be a depolymerization of microtubule, tubulin subunits will be released. Non-kinetochore microtubules from opposite poles overlap and push against each other, elongating the cell. In telophase, genetically identical daughter nuclei form at opposite ends of the cell. And cytokinesis begins during anaphase or telophase, and the spindle eventually disassembles. A closer look for cytokinesis. In animal cells, cytokinesis occurs by a process known as cleavage, forming a cleavage furrow. In plant cells, a cell plate forms during cytokinesis. And on this diagram, you can see on these two diagrams, actually figure A shows us a cleavage of an animal cell in scanning electron micrograph. So So where cleavage furrow is formed from contractile rings of microfilaments, and as a result, two daughter cells form. And in figure B, a cell plate formation in a plant cell, shown by transmission electron microscope here, and everything shown schematically. First, as you notice, vesicles form, which further form plate. And by the way, walls of parent cell, that's a wall of parent cell, and that's actually a forming vesicles, which further transform into cell plate. Cell plate then fully enclose and further these two structures, which before they were one cell, they divide by forming two daughter cells. Here on this slide you can see same event by light microscope. So here you can see nucleus with nucleolus and condensing chromatin. So chromosomes here, and stage prophase, second stage is prometaphase, third stage is metaphase, then anaphase and telophase. Telophase start by forming a future cell plate, and as a result, during cytokinesis, cell plate fully forms and divides a cell into two daughter cells. Binary fission in bacteria. Prokaryotes, these are bacteria and archaea, reproduce by a type of cell division called binary fission. In a binary fission, the chromosome replicates beginning at the origin of replication, and the two daughter chromosomes actively move apart. The plasma membrane pinches inward, dividing the cell into two. And here on this slide you can see a binary fission, stage of binary fission. First, chromosome replication begins. So here is origin of replication. Cell wall, actually all structural features of a Escherichia coli bacteria are shown here. And here you can see two copies of original form. So further they divide, replication continues, then replication when it finish, two daughter cells start to divide, and as a result we can see the formation of two daughter cells of Escherichia coli bacteria. The evolution of mitosis. Since prokaryotes evolved before eukaryotes, mitosis probably evolved from binary fission. A certain protists exhibit types of cell division that seem intermediate between binary fission and mitosis. Here on this figure we can see different types of division in different microscopic structures. For example, the division of bacteria. It goes on by binary fission. Dinoflagellates, they are almost nucleated cells. So they have chromosomes, microtubules, and nucleus during division will be intact. So their nucleus also divide and form separate nuclei of two daughter cells. Diatoms and some yeasts, they also have intact nuclear envelope which will be intact or which will be not destroyed during cell division. But in most eukaryotes, as you can see, their nucleus is formed from fragments of nuclear envelope further, and they also have kinetochore microtubules and plus centrosomes. The eukaryotic cell cycle is regulated by a molecular control system The frequency of cell division varies with the type of cell. These differences result from regulation at the molecular level. Cancer cells manage to escape the usual controls of the cell cycle. Evidence of cytoplasmic signals. The cell cycle appears to be driven by specific chemical signals present in the cytoplasm. Some evidence for this hypothesis comes from experiments in which cultured mammalian cells at different phases of the cell cycle were fused to form a single cell with two nuclei, and is shown in this experiment. Experiment number 1. When a cell in the S phase was fused with a cell in G1 phase, so this cell is in S phase, this is in G1 phase, the G1 nucleus immediately entered the S phase and DNA was synthesized. So, as a result, you can see it here. And in second experiment, when a cell in the M phase was fused with a cell in G1, M phase and G1, the G1 nucleus immediately began mitosis before its proper time. A spindle formed and chromatin condensed, even though the chromosome had not been duplicated. So here you can see one cell has a full number of chromosomes and second cell is haploid. The cell cycle control system. The sequential events of the cell cycle are directed by a distinct cell cycle control system, which is similar to a clock. The cell cycle control system is regulated by both internal and external controls, and the clock has specific checkpoints where the cell cycle stops until a go-ahead signal is received. So here you can see the checkpoints. Checkpoints are important for the controlling of the cell cycle. So for example, here is M checkpoint during mitosis, here is G1 checkpoint, and here is G2 checkpoints. And these are control system of a cell division. For many cells, the G1 checkpoint seems to be the most important. If a cell receives a go-ahead signal at the G1 checkpoint, it will usually complete the S, G2, and M phases and divide. If the cell does not receive the go-ahead signal, it will exit the cycle, switching into a non-dividing state called the G0 phase. So here is G1 checkpoint. A cell receives a go-ahead signal and it continues its division or usual stage of division. If cell does not receive a go-ahead signal, it will not divide. The cell cycle clock. Cyclins and cyclin-dependent kinases. Two types of regulatory proteins are involved in the cell cycle control: cyclins and cyclin-dependent kinases, shortly abbreviated as CDKs. CDKs activity fluctuates during the cell cycle because it's controlled by cyclins. So, named because their concentrations vary with the cell cycle. MPF, for maturation promoting factor, is a cyclin CDK complex that triggers a cell's passage past the G2 checkpoint into the M phase. So here you can see MPF activity and cyclin concentration. If the concentration will be high, so the cell will pass the G1, then S, G2 phase, then mitosis, one more time begins, and etc. A fluctuation of MPF activity and cyclin concentration during the cell cycle. So, during the cell cycle, during interphase, cyclin accumulates. Then MPF activates. Then cyclin is degraded, CDK is formed, and it will lead to the continuum of cell division. So that's molecular mechanism that help to regulate the cell cycle. Stop and go signs, internal and external signals at the checkpoints. An example of an internal signal is that kinetochores not attached to spindle microtubules send a molecular signal that delays anaphase. Some external signals are growth factors, proteins released by certain cells that stimulate other cells to divide. For example, platelet-derived growth factor, PDGF, abbreviated, stimulates the division of a human fibroblast cell in culture. First, a sample of human connective tissue is cut up into small pieces by scalpels, in petri dish of course. Second, enzymes digest the extracellular matrix, resulting in a suspension of free fibroblasts. Third step, cells are transferred to culture vessels. And fourth step, PDGF is added to half the vessels with PDGF, and this is without PDGF. So, the culture where PDGF were added, culture of cells, culture of fibroblasts, they start to divide. A clear example of external signals is density-dependent inhibition, in which crowded cells stop dividing. Most animal cells also exhibit encouraged dependence, in which they must be attached to a substratum in order to divide. Cancer cells exhibit neither density-dependent inhibition nor encouraged dependence. So here you can see on top figure encouraged dependence and density-dependent inhibition and density-dependent also inhibition. Then we can see the growth of normal mammalian cells. And here you can see cancer cells. Cancer cells divide without any control. Loss of cell cycle controls in cancer cells. Cancer cells do not respond normally to the body's control mechanisms. Cancer cells may not need growth factors to grow and divide. They may make their own growth factor. They may convey a growth factor signal without the presence of the growth factor, and they may have an abnormal cell cycle control system. A normal cell is converted to a cancerous cell by a process called transformation. Cancer cells that are not eliminated by the immune system form tumors and masses of abnormal cells within otherwise normal tissue. If abnormal cells remain at the original site, the lump is called a benign tumor. Malignant tumors invade surrounding tissues and can metastasize, exporting cancer cells to other parts of the body, where they may form additional tumors. So on this figure you can see that the primary tumor which appeared in mammary gland. First step, a tumor grows from a single cancer cell, and cancer cells invade neighboring tissue, which is called metastasis. Cancer cells spread through lymph and blood vessels to other parts of the body by blood vessels and lymph. First of all, lymph nodes will be metastasized, or lymph nodes of adjacent or neighboring regions of the body. Cancer cells may survive and establish a new tumor in another part of the body.